Welcome to the final Mini Science 2018 lecture in our series entitled Women in Science at McGill. As you all know, this series is made possible through the generous support of Dr. Lauren Trottier. McGill University is located on land which has long served as a site of meeting and exchange amongst Indigenous peoples, including the Haudenosaunee and the Anishinaabe nations. McGill honors, recognizes, and respects these nations as the traditional stewards of the lands and waters on which we meet today. Many of you have asked us about the videotaping situation, so I'd like to explain that they will be ready on YouTube in about 2 weeks' time. You will be able to find them if you Google the name McGill Mini Science 2018. I will also make sure the link to that YouTube site is posted on the website for Mini Science, so you should be able to find it. I've looked at a few of them, some of the drafts, and other than when I do this and step into they're really well videotaped by McGill Communications, and we're proud that we were able to do that and archive this series in that way. Special thanks tonight to all our volunteers and our hosts who have helped throughout the entire series, but especially to the students from the RMS who've really stood by me when I started to plan this. Throughout the ideas, they got the volunteers for us. And tonight, one of our volunteers is actually helping us on her birthday. And we have a special thank you for her, so she can feel good about it being her birthday, and she has to sit here, pass it back to Noemi Shepherd. If you have not already done so, please turn off your cell phones, Blackberries, or any other devices that may disturb the audience. Tonight you would have picked up 2 evaluation forms. One is for the overall series. Please respond to that one for sure. I have to meet tomorrow with the people in the Dean of Science's office. We have to talk about this series, what we're going to do, how we'll change it, modify it, or maybe put it on suspension for a little bit. I really would like your feedback on that one. And of course, the one for tonight is Anna Hargreaves' evaluation. Our moderator tonight is Heather McShane, the Program Director and Catalyst-in-Chief for the McGill's Sustainability Systems Initiative, which has the best acronym at McGill, that's MISSY. Heather gained a degree in plant science and a master's degree in soil science from Aberdeen University, and then worked for 20 years as a project manager in the information technology sector. She then returned to education to study for her doctorate in environmental chemistry at McGill's McDonald campus, and since 2016 has headed the McGill Sustainability Systems Initiative. Missy is building a community of sustainability researchers from across McGill, from natural sciences, engineering to humanities, management, and law, and supports research at McGill that takes an interdisciplinary, holistic, and solutions-focused approach to sustainability. This is Heather's second turn as moderator. Some of you asked me to please make a note of all the moderators in this series, which I have not done because I corralled them later on as the series was settled. I will make sure moderator names, at the very least, titles, and a brief introduction get posted on and archived on the Mini Science website for this year. Heather, will you please introduce our speaker?
Right. So feeling very relaxed about this. So hello again, everybody. So tonight it's my pleasure to introduce Anna Hargreaves, who's an evolutionary ecologist in the Department of Biology. So Anna arrived 2 years ago? A year and a half ago, yes, in January from UBC, but we shouldn't feel sorry because she's actually from Ontario, so that's okay. She studies, she has 3 main facets of her research: ecology and evolution of range limits, which includes experimental test of range limit theory. I don't actually know what that means, so I hope you're going to explain that. And dispersal evolution of and range shifts. She also looks at geographic importance of species interactions and pollination ecology. She got her BSc in biology and evolutionary science at Trent, where I have to say I was really impressed. She co-designed a field course in ecology and evolution of the Galapagos. I did things with hydrochloric acid and soils in a lab, so. Okay, oh, well, there you go. So did you go on that 14-day field trip?
Oh, I but not as an undergrad.
Okay, fair enough. I was really impressed. After she did her bachelor's, she studied for 2 years, actually doing her honours thesis between Trent and the University of KwaZulu, Natal in South Africa, looking at plant pollination systems. And she did a year on bird research, which took her to Nunavut and to Costa Rica. She then went on to do a master's degree in Ecology and Evolution at Calgary University. And then she took another break to work for 2 years in the Centre for Conservation Science at Calgary Zoo, which her fieldwork was in Alberta and this time in Ghana. She then finally did her PhD in Ecology and Evolution at Queen's University, where she was awarded the Governor-General's Gold Medal Award for her thesis. She moved from there to University of British Columbia, where she took a 6-month mat leave and then went straight back to work. So these are some of the awards here. I'm not going to read them all out, but I'd just like to draw your attention to some. She recently won, or was awarded, the American Society of Naturalists Young Investigator Award. She has the New Phytologist Award for Outstanding Student Presentation in Botany twice. She got the Queen Elizabeth II Scholarship in Science and Technology, the Queen's R.S. McLaughlin Fellowship, the Queen's Tri-Council Recipient Award, and I could go on. And interestingly, and maybe we could ask her a question about this, she got the Commonwealth Scholarship, or was given a Commonwealth Scholarship for the University of Canterbury in New Zealand and turned it down. So I'd like to know why. So with that, I'm really looking forward to this talk, and I'd like you to help me to introduce her. Thank you.
Thank you, Ingrid, for inviting me and organizing this, and Heather for that introduction. And thank you to all of you for coming. Before I go any further, I just want to brag a little bit and say that Noemi, whose birthday it is, was also working in my lab this year and just handed in her independent research project. And so I know that this is the last in the series where you've heard a lot about science at McGill and women in science, and I know that you've heard from some really interesting and dynamic speakers, and I think probably it's safe to say that each of us have our own take on what this theme means. And so today what I want to talk about is, I've chosen this theme of diversity because it forms a really beautiful bridge, I think, between the two core elements of this year's theme. And so I'm interpreting women broadly as diversity in STEM, so gender diversity, but other kinds of diversity as well, and just thinking about diversity in terms of our human interactions. But then also, diversity is at the heart of science, and especially of biology. And so I have multiple goals for this talk. The first is just to get you jazzed about how awesome the natural world is and how cool it is to study. And this is because of the endless diversity that we find there. The second, I want to convey to you the beauty and the excitement of the detective story of science. So the scientific approach that I take really does capitalize on this sort of mystery-solving, question-asking, evidence-gathering way of doing science, which I think is really fun, and maybe we don't think about enough when we think about science, especially when we think that science is dry. And then I want to draw parallels between the scientific method and the way that we study diversity in nature, and how we can think about sort of diversity and equity issues in our human relations, and the sort of day-to-day human interactions that we all engage in. And then sort of the fourth and last but not least goal is to share with you some of the projects that I've been involved in. And then one more small piece of bragging, which is that the woman depicted as the logo of this year's Mini Science is not just a made-up cartoon. She is a real person, Sandra Clement-Gasson, who was my very, very first student. And she did an amazing job trying to start up a system to look at bacterial evolution, which I will not talk about at all today because they are totally not photogenic and it didn't really work. So, like physics or chemistry, biology is a huge field, and I am going to talk about just my small corner of it and ignore the vast majority. So we are not going to talk about medicine, we are not going to talk about physiology, molecular biology, and genetics, even though they are all interesting in their ways. So my corner of biology is ecology and evolution. And ecology and evolution really is the study of diversity. So classic questions are, why are there so many species on Earth? Why do we have so many bees? Why do they look different? Why do we have so many wasps and crazy caddisflies? Why are there so many fungi? What can we learn about them from looking at how they're different and studying how they're different? What does that variation mean between species? What does it mean for how these species interact? Below the level of species, there's tons of individual variation. So just as humans, we're one species, but wildly diverse, the same goes for other species and other levels of organization. And this has really interesting implications. How does that variation between individuals affect how they interact with each other? How does it affect their success in growing, in surviving, in finding a mate, in reproducing? And then how does that variation in success between individuals lead to the creation of further variation through adaptation and evolution? And these ideas of diversity, I mean, you can type up diversity biology art and you'll find a ton of illustrations like this that show that this idea of variation in the natural world captures not just the imagination of scientists, but also artists and poets. A small pop quiz. Does anybody know who the artist behind these beautiful illustrations is?
Is it Durr?
Nope. It is someone I guarantee you have all heard of, probably one of the most famous writers and illustrators.
European or North American?
But I don't know. English, I think.
English.
Beatrice Potter.
Oh, wow.
Yeah, so in case you didn't know, not only does she do beautiful illustrations in Peter Rabbit, but she actually has an entire book on les champignons full of stunning watercolor illustration, and she was a really accomplished natural historian. Is it an answering message for me? It's right. Okay, it's the perfect time to drink some water.
Yeah.
And actually, this is a great time for me to ask you, because I want to make sure I'm not over-explaining things you all know about. How many of you would classify yourself now or at any part in your life as a scientist or doing science? Okay, so I am preaching to the choir about the beauty of science. How many as biologists? How many of you have ever done research? Great. Okay, ecology, evolution? Okay. Super. So then I want you to think about what I'm telling you, not as like new information that you're receiving, but maybe a new way of thinking about the implications for how we study diversity and how we think about diversity in day-to-day life. So here is a member of a group that is a classic example of biological diversity. Any ornithologists among us? Bird of paradise. So this is a bird of paradise. This is actually the superb bird of paradise, as you would guess. And just looking at this bird, I think would lead most people to think, what on earth is it doing? Why does it look like that? Is it doing it any good? Is it like, I tried to download this on video, and I won't act it out, but I did not successfully get it on video. But this is just one freeze frame of an elaborate dance that these birds do that differs dramatically among species. And they come with these wild, elaborate plumages and these weirdo sort of dances and displays. And they're one of the most famous examples that people like me, when we start off talks, like to use to sort of show how interesting and seemingly weird and elaborate diversity in nature can be. Interestingly, all of these birds that we look at share something in common. They are all males. And so if you actually look at female birds of paradise, usually in these sorts of presentations, we only show pictures of males. And it's the males that have the obvious visual diversity. They have this elaborate plumage, these crazy dances. Females are just as diverse, but they are diverse in a very different way. They're diverse in their assessment and their decision-making and their ability to identify the traits they're looking for in a mate and to assess the different potential quality of the birds that are displaying to them. And we don't often appreciate this, and this leads to my first small lesson that we can learn from, apply from sort of the natural world to our own lives, which is that we can't learn anything if we just focus on one subset. So if we only look at male birds of paradise, we'll never get the full picture, which is doubly complex. It's complex not just in plumage, but in decision making. And this is equally true across human affairs. So whether everyone's thinking of testing different pharmaceuticals or looking at predicting the uptake of new technology, or how market economies function, if we just look at a subset of the individuals that they affect, then we don't get the whole picture. And usually, when we look at a subset of individuals, we all tend to look at the people that look like us. And that's just an innate human way of thinking. And so it's especially, not only do you have to think about who you're considering, but you have to check your own biases about who's out there and what's normal. So while I have never had the privilege of seeing birds of paradise in the wild, I have, as Heather mentioned, worked with birds. And so this is a photo that I took up in the Canadian Arctic. No, Amy, do you know what? So these are red phalaropes, and they are charming, funny, hilarious, not super sharp little shorebirds, and they form nesting pairs and they raise their chicks on the tundra. What's interesting about these birds is that this one is the female. And so for these birds, it's the female that has the brighter plumage. It's the female that has multiple mates. She does, of course, lay the eggs because that's the definition of being female. But the male incubates them, and he does all the parental care. And so here's our second small take-home lesson, which is that there's always exceptions. And so anytime you hear some rule about anything biological, you should have a high degree of skepticism. And often the exceptions are really interesting. And I think there's a lesson here to to avoid thinking of things in black and white. So another interesting part of the human brain is that we really like categories, and that's as true of scientists as anybody else. So you can just look at taxonomy to see how much people like grouping things together. But scientists are also, for the most part, or at least we try or should try to avoid thinking of things as black and white and to avoid thinking of things as having concrete answers. So scientists, especially biologists, live in a world of probabilities. And you can really see this if you've ever heard a media interview with an ecologist and how slippery they are when the interviewer is trying to get them to say, you know, what's going to happen, or does that mean this? And the scientist will like deke and weave to be like, well, maybe, you know, or in these set of circumstances, or to the best of my knowledge, or I really can't say. And this makes them, I think, super annoying to reporters, but I feel like the world could use a little bit more of this perspective. So few questions that matter in the world have definitive answers, and this is just as true in human society as it is in biology. And I think we could go far by embracing uncertainty and nuance. So the second way so this study was on breeding bird success, but the way that I started studying birds, or first did my own research on birds at least, was from a very different perspective. And it was looking at their relationships to plants. So this stunning photo was taken by a friend of mine named Dale, who now works for Swarovski and takes amazing pictures like this. But when I met him, he was a biologist. And this is a spider hunter in Thailand. And it's clearly feeding on this beautiful, astounding, eye-catching flower. And so here are the individual flowers. And you can see, for any of you who need a refresher on floral biology, and I promise to use the mouse to do things like this. Here we go. So there's the flower. Protruding from the mouth of the flower are anthers. And anthers are the organs of the plant that have the pollen. So this spider eater is drinking nectar from this plant and potentially pollinating these extraordinary flowers. And so pollination relationships are, I think, an especially cool thing to study among all of the cool things to study, as you have these two totally unrelated species that come together in this interaction. And so plants are, they don't move, obviously, and but a lot of them need to mate with another plant, a genetically distinct individual. And to do this, they have to court not another individual of the same species, like the birds of paradise, and you can already see how much work that is, but they have to court an entirely different species. So you have to somehow attract this totally different species who doesn't care at all about your reproduction and manipulate it in such a way that it will take your gametes to the next flower and bring gametes to you. And so this is quite a complicated and you know, at face value, a bit weird interaction. And these types of pollinating interactions have resulted in some of the extraordinary diversity that we see between flowers, but also some of the extraordinary diversity we see in birds. So these long, thin bills shared by spider hunters and hummingbirds and sunbirds in Africa, for example, are adaptations to drinking nectar out of flowers. So you may have noticed that I said maybe pollinating, and to many people that seems funny. So you see this animal, it's clearly visiting the flower, and you would maybe just call it a pollinator at face value. But it turns out it's not enough. Pollinating a flower is about more than just visiting the flower. And so there are actually tons of animals that visit flowers, and only some of those pollinate. So if we look again at this spider hunter, you can see that it has clearly, and here in this plant you can actually see that the pollen is white. This is the pollen coming out of the anther. And you can see that the bird has picked up a ton of pollen on its bill. But you'll also notice that the pollen stops here. So it's not reaching the feathers of the bird, and it's not even reaching these sort of hairs at the base of the bill. And so it's possible that this bird would carry this pollen to the next flower. But birds are really good at grooming bills, and bills are not very sticky for pollen. And so the bill, especially a long bill like this, is actually not a great place to put pollen if you want to get it from flower A to flower B. So whether or not these birds pollinate is uncertain, but what we know for sure is that they've consumed nectar. So you can see this little bird has its tongue out, which is usually a really good way of telling that either a bird or an insect has just drunk nectar. And when an animal, so we think of, because humans are motivated by the market economy, pollination is often expressed in sort of market economy terms. And so the idea is that a visitor comes and gets reward, and it pays for that reward by pollinating. And so then the converse is that if a visitor comes and takes a reward and doesn't pay for it, it's a thief. So there is a whole body of literature on animals that steal what are supposed to be legitimately paid for rewards. So that's nectar and pollen. And a lot of my early work was on pollen theft in particular. And pollen theft is especially important because if you steal pollen, you're not just taking a reward that a legitimate pollinator could have taken, but pollen, of course, is essentially the sperm for plants. So you're also removing gametes, which has potentially a direct reproductive cost to that plant. So if one knows about this, you can then look at a whole bunch of features of flowers that initially seem designed to attract certain pollinators, and many of those features can be reinterpreted as trying to avoid or repel potential thieves. Interestingly, so when we reviewed the literature and all of the accounts of pollen theft, the most common pollen thief by far was the honeybee. You can ask me more about that at the end if you want. But it's really interesting because it's probably the thing that we classically think of as augmenting pollen. And so there's lots of agricultural and some community conservation programs that operate under the assumption that honeybees are good pollinators. So my work on pollen theft was in South Africa, and South Africa is a pollination biologist's dream. It has incredible floral diversity and incredible diversity among pollination systems. And this project, like most of my work, involved a combination of synthesis, so the way that we found out that honeybees were the most common thieves is by going to all of the literature and gathering it all together and analyzing the data together. But the other pillar of my work is field biology. And so this is not the only way to do ecology and evolution, and I was going to show you a page of just code that just to confess that even though all I'm going to show you are stunning pictures of the field, actually the majority of my time is in front of a computer, but I still consider myself a field biologist. And so it's not the only way to do ecology and evolution, but it's my favorite. And it comes with a long and noble history of biologists tromping into the wilderness, trying to learn about nature. Through being in and manipulating and observing the natural world. One of the, I would say the world's most famous field biologist is Jane Goodall. I cannot think of any, not even field biologists, but few biologists have the sort of name recognition that Jane Goodall does. And her work beautifully illustrates the first two parts of the scientific method, which are curiosity and observation. And so observation, when we talk about observation in terms of science, it's not just seeing. Observation is an active process driven by curiosity, and it's the type of seeing that allows you to notice things that are interesting. So it requires that you have some sort of framework for what is normal or expected so that you can recognize exceptions. It requires you to ask why things are the way they are. Know the story of the apple falling from the tree that led to the discovery of gravity. How many people had seen apples falling from trees before, right? It's not seeing. It's the connection that's the questioning behind the observation. And I think that, as an aside, when we teach children or undergraduate students about science, this is the part of science that we most often fail to convey adequately, is the importance of curiosity and observation and active observation and interest in the natural, well, in anything. In my case, in the natural world, but the same could be said in any of the sciences. So Jane Goodall's most famous discovery, anyone on a hazard? What discovery do you know Jane Goodall for? Tool use in chimpanzees, exactly. So this was groundbreaking, partly because it was just groundbreaking, but partly because of our total human-centric bias in how we see the world. So at the time that she was doing this research, so humans love to talk about what makes us different from everything else. And we love to, to do this in sort of an absolute statement, like humans are the only animal to do X. And at the time that Jane Goodall was doing her research, one of the common ways that that statement was made is humans are the only animal to use tools. And so the cultural impact of her discovery was not just that, oh, this is awesome, chimpanzees use tools, we didn't know that, but it shook the foundation of what we thought it was to be human at the time. Since then, we've just moved the goalposts. So now lots of things use tools, it turns out. So that's no longer a good way of, no longer regarded as important. Now it's other things like, oh, there's so many of them, to laugh. And I would say a small lesson here is that the next time, or if ever you hear anybody start to make a statement like humans are the only animals too, you should just roll your eyes and stop listening, because it's probably not true. And even if it is true, it's, potentially there's some elephant saying elephants are the only animals to drink water through our noses and squirt it up in the air, and this obviously makes us superior to all other life forms. So we're very biased about the type of statements that we make. They're always designed to put us at the top of the tree of life. And so Jane Goodall's discovery led to a beautiful quote by her collaborator, Louis Leakey, which is that now we must redefine tool, redefine man, or accept chimpanzees as human. And this quote, I think, obviously we didn't do any of these things, but it illustrates one of the important parts of the process of scientific discovery, which is that you have to have a way of incorporating new knowledge into your worldview. And how do you change a paradigm? How do you incorporate new knowledge in? And I think this is a lesson that science has to teach other forms of thinking, perhaps, is that there's value to uncertainty and to not knowing, and there's value to changing your mind. So I'm not going to get this quote right, but there's a politician who said something along the lines of, he was accused of changing his mind and waffling on an issue, and he said, when I'm presented new evidence that counters my old worldview, I change my mind. What do you do? And I think that this attitude, obviously we want to make sure that the evidence is good and we want to allow it to accumulate, but ultimately all of our ideas should be subject to new evidence, and we should try and maintain that flexibility in thinking where we don't become so attached to what we think we know. So back to fieldwork in South Africa. So I worked with a group of plants called aloes. You probably know aloes through aloe vera, which is the most famous member, and probably lots of you have broken a leaf of aloe vera at some point and smeared it on some burn that you wish you didn't have. But what you wouldn't know necessarily, and what I had no idea about looking at these like tiny plants of aloe vera that we see in Canada, is that in their natural environment, aloes are stunningly beautiful. They can be trees, so there are tree aloes that are the size of a maple tree, and they're incredibly diverse in form. So you get aloes from tree aloes to this big. They have different flower rays, and they have these huge inflorescences of flowers. And when I started, so I had done my honors thesis in South Africa with a different group of plants, and when I went back to do more research, even though aloes are this iconic South African species, South African group, there had been very little work done looking at their pollination biology. And so we used this group of plants to really look at pollen theft from a few angles. And I'm only going to tell you about one study. And I'm going to pick this one because I think it's a really nice illustration of the scientific method, which it turns out you all know about anyway. So it'll be a fun detective game, I think. So actually, and before we do that, I want you to look carefully at this photo plate. So we're going to test your powers of observation. Look carefully at this photo plate, and I'm going to show you the aloe that this research story is about, and I want you to see if you can spot the differences. So here's the smattering of aloes. These, I would say, are what all 350-ish of about 354 species look like. The other 4 look like this. So this is Aloe rhyhydensis. You can see how stunning this part of South Africa is. It's sort of up in the northwest corner, just below Kruger National Park. And when we zoom into the flowers, this is what they look like. So there's 2 unusual things about these flowers. One is unusual for aloes, and one is just weird in general. Any guesses or observations about what they are? I'll give you a minute. Okay.
There were 2 different kinds of petals or whatever.
The things sticking out?
Yeah.
Yeah, so these are, come mouse, here we go. So these are the anthers, where the pollen is, and then these are the stigmas, so this is the male part of the flower that produces the pollen, and this is the female part of the flower that receives the pollen. And so it's true that this is a bottle brush type of aloe where those are really exerted. So that is very weird compared to Canadian plants. It's not as weird for aloes. But good breaking the ice. Anybody else? Mm-hmm.
Nothing about that liquid, that pool.
What about it?
I don't see that common, like such a large pool of nectar or whatever it is.
Yep.
The base of such a tight flower?
Yep. Yeah. So it's weird to be able to see nectar. And there's something else that's weird about the nectar, which maybe is not as obvious because it's so hard to see nectar.
Is it a jelly?
No, it just kind of looked like a jelly. No, it actually is a normal liquid.
It looks like blood, or it looks like a...
The color. The color is super weird. So this plant has black nectar, which is incredibly rare among plants. So it's not just, there's 4 species of aloes that have black nectar, and there are other sort of sporadic accounts of black nectar from a variety of taxa, but it's really, really unusual. Nectar is usually a clear, colorless liquid. The other thing, I'll just tell you this one because you guys have been good sports. The other thing that's weird about this aloe is that whereas most aloe flowers are tubular, so you can see varying lengths, so a little flared, open, but still tubular, short tubular, longer tubular and hanging down, but you see the theme. These ones are quite open. And so the flower itself is open, and then the anthers and the stigmas sort of follow that line of the corolla and splay outwards. So I was really interested in studying, so part of the project was that we were doing a survey of species with different flower shapes and different kind of floral structures to try and explain why some were pollinated by birds and some were pollinated by bees. And I was really interested in this species because of the weird flare of the flower of the corolla. But one of my collaborators was, who had a much deeper history of pollination biology behind him, recognized how unusual it was that the nectar was black. So we were both interested in unusual features of this plant, but different ones. And so we went in knowing that the plant was a bit of an oddball. And then there's a suite of things that you do as a pollination biologist, and so you take some flower measurements, and one of the things you usually do is you measure the nectar, and you look at its sugar concentration. And so you do this with little tiny microcapillary tubes, and you suck up the nectar, and then you blow it back out onto this lens, and it tells you the sugar concentration. And field biologists are not known for being meticulous, This is why I'm a field biologist, and I would be a terrible microbiologist. But inevitably, as you do this and you're going too quickly, you get some nectar in your mouth. Usually, it's not a big deal. This nectar was noticeably bitter when we tasted it. So now we have nectar that's black, and we have nectar that's bitter. And bitter nectar is even more weird than black nectar, because the whole point of nectar is that it's an attractant. Right? So the whole raison d'etre of nectar is to be consumed. It's one of the few plant products that are actually meant to be consumed. The only other one is fruits. Not the only other one. The only other common one is fruits. And so why have bitter nectar? I'll leave you to ponder that question for a minute.
Obviously to repel, to deter the birds or the insects.
Exactly. So this is what we were thinking. So potentially, so this is one idea. This is the coolest idea that we'd thought of. But one thing that is a good practice in science is to have multiple hypotheses at the same time. And the reason for this is to make sure that you've fully fleshed out the options, but also to avoid the very human instinct of getting attached to your own ideas. So if you're competing, then you're less likely to be on the side of one versus the other. So another option is aloes are... there's the succulent component of the leaf sap that is soothing, but there's also a lot of what are called secondary compounds. And so interesting chemistry within aloes. And it's possible that that sort of leaches into the nectar passively and has no biological purpose within the nectar itself. So that's another option. It's also possible that it's the color somehow is either a repellent to visitors that you don't want or an attractant to visitors that you do want. So the second observation, so we had this observation that the nectar is black, we knew coming in, that it's bitter, which we didn't know, and then we also noticed that although so in South Africa there's no hummingbirds, but the ecological equivalent look a lot like the spider hunter that we looked at earlier. They're called sunbirds. And sunbirds are voracious feeders on aloe, many species. But even though there were tons of sunbirds in the area, we didn't see any landing on this species, even though there's obviously a ton of nectar being produced. So what we did see on this species were these short-billed generalist birds. So this is a bulbul, and you can see that its bill is quite short and stout, and this is a white-eye, and again, you can't see the bill very well, but it's quite short. It's like a nuthatch type bill. And so we thought, okay, well, these birds, and with their bills, seem to be a much better fit to this flower. So if you line them up, this is what the flower looks like. This is sort of an example of a short-billed bird that we saw feeding on it. And you can see the orange pollen stuck to this bird's feathers. And so as we mentioned before, bills are not a great place to put pollen. And so this is a classic sunbird shown to scale. And you can see that if the pollen did get on it, it would be only on the bill. But then also because these anthers are splayed, they're not well positioned to contact that bill. So this bird could easily access the nectar without getting any pollen on it. So we have our hypothesis. So we thought, well, maybe dark nectar attracts these generalist birds that are used to looking for things like insects or seeds, which are kind of dark and shiny. And we thought, okay, well, dark insects and seeds are also bitter. We assume. So maybe they're just not bothered by the bitter taste, and so maybe it's a passive thing that doesn't really do much either way, or maybe it's a repellent to sunbirds. Okay, well, I'm over time, so I'm not going to make you guess how we tested it. So the way we tested it is we made these fake flowers. And so we just cut the ends off of Eppendorf tubes and we taped them. So we had one taped with yellow tape, and the tape is on the outside, so it doesn't affect the nectar. And we had one with black tape surrounded by yellow tape. And we offered these to sunbirds and to 2 species of short-billed birds, the Cape White-eye and the bulbul that I showed you earlier. And it turns out that neither of them care about the black center, so they were just as likely to go for one as the other. So we could rule out that hypothesis. So the black nectar doesn't seem to be having either a visual attraction or a visual repelling function. So then we filled them with just a sugar solution of the same concentration of sugar or the actual nectar, and we did the same choice experiment. And again, there was no difference in which the bird went for initially. The short-billed birds, whichever one they went to first, they drank all the nectar, and then they went to the next one and drank all the nectar every time. The long-billed sunbirds, so they chose randomly. If they chose the nectar one, they were happy, they drank the whole thing. If they chose the sugar solution. If they chose the dark nectar, it was classic. They would pull their head out of the nectar, they would shake it, and they would fly to the other end of the cage and sulk. And so it had, and they never finished drinking it. So they would eventually come back and explore the sugar solution, and they would maybe drink it, but they never finished drinking the small amounts of this bitter nectar that we put in. And so sunbirds, as I said, consume a ton of nectar, and so we don't know the steps of this process. Was the bitter nectar first, and then the flower shape evolved, or did the flower shape evolve, and then the plant sort of leaked in increasing amounts of phenolics? But what we do know is that it has no attractant function, but that it's one of the few examples where we can say definitively that it's a feature designed to repel certain flower visitors. Okay, I'm going to skip that one. The last thing, I wanted to just talk really briefly about work that I'm doing now. And this was inspired, so I love pollination biology, but at some point I wanted to think about interactions between species in a broader context. So we know that they affect the performance of individuals. We know that they're important from an evolutionary perspective. We know they can have some influences on populations. But at a big global biogeographic perspective, how important are they? Do they determine species distributions? For example, and so there's a long-standing idea in ecology and evolution and biogeography that we can predict where interactions will be really important using what's illustrated here, which is the gradient of species diversity. And so here, this is illustrating essentially a heat map of the number of species, and the warm colors are high diversity, the cool colors are low diversity, and it's beautifully illustrated for the northern hemisphere and our part of the northern hemisphere, but the same applies in sort of Europe and Eurasia and in the southern hemispheres as well. So species diversity increases dramatically towards the equator fairly universally. And it seems then intuitive that we might predict that as there are more species interacting with each other, maybe those interactions become more important. And so this, I won't go into all of the ideas that are built upon this hypothesis, but there's some pretty big biogeographic hypotheses that sort of assume an underlying gradient in the importance of interactions between species. But it hadn't been tested experimentally. So during my postdoc at UBC, I started thinking about this and how you would do it. And so testing this type of gradient experimentally, the first obvious challenge is space. You have to get to sites that cover a big enough gradient that it's convincing. But the second challenge is evolution. So if you can't think of anything to ask me during the questions, ask me to explain what's going on in this slide. But plants, when they're chronically exposed to antagonistic interactions, they develop all kinds of defenses. So if you have chronically high herbivory, you would expect plants to evolve mechanical defenses and chemical defenses. So plant chemical defenses are responsible for many of our drugs, like aspirin, many of our maybe medicines like aspirin, drugs like cannabis, opium, spices, lots of the things we love to cook with, and perhaps there's no coincidence that many of our spices are actually tropical in origin. And so what this means is if you just go out and observe interactions, so if you just counted how many leaves were eaten, for example, you wouldn't detect necessarily the underlying pattern because you would have expected plants to evolve their way out of a problem. So you really need to use some sort of standardized material. And so what we did is I started a project using seeds, and we measure essentially post-dispersal predation on seeds. And the beautiful thing about seeds is that they're very stable. So once you have spent hundreds of hours getting around customs forms, you can mail seeds to your collaborators, no matter where they are in the world, and everybody can use identical seeds. So you can get rid of any sort of evolved associations. And so what we did is a really simple experiment where we have what we call a small seed depot. We use sunflowers and oat seeds, and we put out 30 of these depots at each site, and we would just come back and check them 24 hours later. And so it's incredibly simple, and you just score how many have been eaten, how many have been removed as a metric of predation. And if you're really lucky and you work in Vancouver, where I was at the time, you catch charismatic megafauna in action. So this is a banana slug actively eating my seeds. This is the most exciting moment of the whole project, other than submitting the paper, which happened last week. So sometimes we catch herbivores in action, and we did an initial experiment where we put wire cages. I'm going to show you that later. So the experiment is quite simple. The trick is that we did it across this huge geographic scale. So we have 18 latitudes from Alaska all the way down to the equator in Ecuador. And each of these triangles is not just a site where we did it, but it's a mountain where we replicated that experiment going up the mountain. So we have the elevation crossed with latitude, and we can look at their combined effects. I actually, you know what, I just added up 887 hours of my time, plus however much time all of my amazing collaborators spent on it. Yeah, so I put together this network of awesome people. I started by harassing my friends, and then I called their friends, and then in some cases I cold called people that I still haven't met. And so we did this experiment multiple times at most of the transects. And we also did an experiment where we caged some of them. So this allows us to at least separate the effect of invertebrates from the effect of vertebrates. And we thought maybe invertebrates would show stronger patterns because their activity is directly affected by temperature. And I'm not going to show you any graphs, although it turns out for this audience I totally could have. But we found significant increases in seed predation from high latitudes, such as Smithers, BC, to low latitudes, such as Costa Rica, just as predicted. So seed predation increases in intensity towards the equator. Likewise, we found significant increases from high elevations. So this is 4,000 meters in Ecuador to low elevation. So this is almost at sea level also in Ecuador, which is our most outstanding transect. And so so it's totally exciting because it's a small, simple experiment, but done at a big scale that allows you to get at patterns that we would have just never been able to get at otherwise, and that we would probably not have been able to get at easily even a few decades ago before email and better, in theory, postal services. I have some things to say about the Mexican post system that sort of allow this connected networks of scientists working together, which I think is a really exciting model moving into the future that's happening more and more. So the last thing I want to show you is this video. Let's hope this works. From Colombia. So in Colombia, they set out little camera traps, because otherwise we never know for sure that we got mammals, but we busted this little guy eating our seeds. And you can see another one here. Here you got really like, so you can see the sunflower seeds going into this guy's face. And it actually, bloop, it leaves one here. You can, oops, come back. That's a sunflower seed, but it comes back for it. So I'm going to leave it there, but I am totally happy to take any questions about anything we've talked about or anything else you feel like asking. Thanks very much for your attention.
So thank you very much.
You're welcome.
I guess now we all want to go back and study biology.
That's right. That was the main goal.
Well, it was
So-
really inspiring. Yes. So I'd ask you to speak into the microphone, even though you don't hear it, because it will record the video. There you go.
You just mentioned in passing the bird in the cage when you were
For this experiment.
in South Africa.
Oh, yeah.
It was. You said birds in the cage went into. I heard something.
Oh, yes, yes.
How did you?
What was
get them in
happening?
there? What did you?
Oh, okay, good. It took me a long time to figure out what you were talking about. I totally said that.
I was quite sure I heard the
No,
word cage.
I know, we definitely did. So we had, the university I worked in South Africa had this amazing outdoor aviary facility. And so they had some birds that were kept there all the time. And then, but you could also catch birds in the wild and bring them in for a week and do things like this experiment and then let them go. So that was the cage. And at the time, because I was still young then, I thought that this was common. And now I realize that actually very few universities have amazing outdoor aviaries. So it was a really lucky place to land for studying bird pollination, because we did a lot of the detective work we just wouldn't have been able to do otherwise.
Other questions? So I have a question, which is, how could you possibly sit in front of a computer screen after you've done all this?
So this is going to sound, when I was a graduate student, I would hear my advisors say things like, oh, field work, like, that's for graduate students. And I thought, what are you talking about? Why don't you want to go outside? And then I did 20 months of field work for my PhD. And eventually, at first it's like going outside, but you're only looking at the ground if you're studying plants. And you spend your whole time counting plant nodes. And so now I feel like I would still like to go outside, but I would like to go to more than 4 mountains and look up. So, so actually, it's increasingly, I killed my fieldwork itch during my PhD. I never thought it would happen, but now I am happy to accompany grad students and do the classic thing that my supervisor did that I thought was totally annoying, where they say they're going to come out and help you with fieldwork and just take photos the whole time. But then, as soon as you need to make a presentation, you really appreciate that contribution. So that's the role that I'm about to embrace. So,
Will be appreciated.
mm-hmm.
Any other questions? Yes. Yeah.
I have 2, if that's okay. The first one is what was happening with the plant with all the spines or whatever.
Thank you. So, do you want to ask your second question as well? Since
The second one was, you didn't mention what you did your PhD thesis on yet, did you? So I wanted to know what that was too.
Yeah, no, I totally skipped my PhD thesis. Although this photo is from my PhD thesis. So the bridge between the interactions that I talked about in South Africa, which is my master's thesis, and the global distributed experiment, which is my postdoc, was that I was really interested in looking at, and I still am interested in looking at species distributions. And so I got there via the path I described, which was being interested in how often are biotic interactions important in defining species distributions. But then it sort of, so I established a new study system which used steep elevational gradients in the Rocky Mountains in Alberta I used a small annual plant where we could transplant it between elevations in its range to look at whether populations were adapted to the elevation they come from. And we could use it to test whether performance, like survival and reproduction, declined towards the edges, and whether the species would be able to make it beyond its current distribution. And we crossed that with a warming experiment where we used plastic cones that essentially act like mini greenhouses to passively warm the air around some of our transplants across its upper range limit. So we tested the direct role of temperature. There was lots of other experiments in there as well, but so that was the gist. So for my PhD, I was looking at species distributions and particularly interested in the evolutionary and ecology processes at the edge of distributions. And I still am super interested in that, but it just takes longer to explain. So, so I don't tend to talk about it as much in public talks.
So what happened towards the edges?
Sorry?
What happened towards the edges?
So for the high elevation limit and beyond, so if we plant the species above its range, what happens is that the seeds germinate, but plants can't have a really hard time making seeds. And this is largely about temperature and heat accumulation in the growing season. So when we warm those plants, their success increases. And the plants from the high elevations at the edge of the range are have the best success when we plant them beyond. So we do see what's called local adaptation, and it improves their fitness beyond their current distribution. At the lower elevation range limit, its fitness is also really low, but it's the sort of opposite problem. So at the upper limit, plants can't make seeds. At the lower limit, seeds can't make plants. And so we have really, really low emergence. So we planted thousands of seeds and we got 5 plants. My first graduate student is going back to Alberta to try and figure out why that is. And that was how I got into seed predation in the first place. So I did a small pilot study across the lower range limit where, very similar to the one I described, and we found a significant increase in seed predation across those low sites where we had really low emergence. So it's possible that seed predation is playing a role in defining that limit, but we didn't follow seeds through. So what we're going to do now is do some more experiments like the ones I've shown, but then also plant seeds in a way where we can find them if they don't germinate the next spring and figure out how many were eaten versus how many just died.
And I think I've just learned what range limit theory is.
Yeah.
Yeah, great.
Do you want a brief synopsis of this? This is a stinging nettle. So it's actually not a tropical plant, but it's got to be one of the coolest, weirdest plant adaptations. So each of these are enforced with silica. So essentially they're made out of glass. They're hollow, so they're glass tubes, and each one ends with this little bulb, which has, by design, a really weak point here at the base. So when you brush against stinging nettle, the bulb breaks off, and that leaves this jagged, sharp opening, which is essentially, and then at the bottom is this squishy tissue full of this cocktail of irritants and toxins. So you brush against it, the tip breaks off, the spiky glass tube penetrates your skin, and as you push down on it, it pushes into that squishy bit and acts like a needle, a syringe, and it injects you with these skin irritants, which include things like formic acid, which is what are in ants when they bite you. So it's this terrible, beautiful defense system that is, hypodermic needles. Yes.
Absolutely amazing. Thank you. So you really, very quick. Yeah, sure.
I guess I wish I'd brought my daughter because she would have loved this. And I just wondered, do you do outreach at all? Do you talk to schools or anything like that? Because it seems to me this is an amazing,
Yeah, I try. So my outreach policies, I say yes to everything. And then once I've been in a place long enough, I try and establish those connections. But I haven't been in Montreal long enough, but I would love to start it. So yeah, hook me up.
Except remember that if she calls you, she's asking you to do field work.
That's right. If you happen to live on a mountain.
Yeah. So please join me by thanking Anna for a really interesting talk. Good.
That's very
There we
sweet.
go. Do you want to close everything again?
Thanks, Heather. So I guess now's my chance to just thank you all for attending, for being interested, for asking great questions, for appreciating what we learn from women in science at McGill. Keep your eye on the website. We'll see you next year, I hope.
And thanks to Ingrid.
Thank you.